So welcome to the ACAR ANU student presentation session. We acknowledge and celebrate the first Australians on whose traditional lands we meet and pay our respects to elders past and present. Today I'm currently on Dharawal Nation land, and I encourage everyone to think about whose land they are on. So my name is Molly Turnbull and I'll be chairing today's session. Our first presenter for today is Amy, and she has her lovely poster ready to go. So Amy, take it away when you're ready.
Thanks for the introduction, Molly. Like Molly said, my name's Amy and I'm a student at the University of Adelaide. Today I'll be presenting my research that I did during the third year while I was studying my Bachelor of Science, and it's about the Kangaroo Island echidnas and the recent bushfire season they experienced. So just to start off, I thought I'd give everyone a bit of background information. So in 2019 to 2020, Australia experienced its most devastating bushfire season yet. These fires have had a huge impact on the plant and animal biodiversity, food availability and habitat. So Kangaroo Island is the third largest island in Australia, and during the bushfire season, almost half of its landscape was directly impacted by the fires. Kangaroo Island is home to one of 5 short-beaked echidna subspecies who were recently recognised by the Australian government as endangered due to habitat destruction, roadkill and invasive predators. So the bushfires really added to the pre-existing threats and pressures that these local echidnas already face. And so part of my research will be to explore the impact of the bushfires on their health and biology, specifically using the microbiome as an indicator. So for those that don't know, the microbiome refers to the microbial population like bacteria, fungi and viruses and their combined genetic data within an environment. So the gut microbiome has become quite widely well known as a good indicator of health and well-being, particularly with human diseases. It's already started to help us understand asthma a bit more. It's currently being employed in animal studies as well, so looking at wild populations to then inform conservation efforts. So echidnas are one of Australia's most iconic animals, and because they're also one of the oldest living animals in Australia, they're quite generally well adapted to fire. So they have been documented to dig underground while there are bushfires going on, and so they can kind of go into this hibernation-like state called torpor until it's safe to re-emerge from the fire. On top of this, their foraging behaviours tend to change as well depending on the intensity of fires. So if it's a more low intensity fire where a lot of the original habitat is still intact, echidnas are still able to roam, still able to forage quite easily. But in the case where the fire intensity is a lot higher, a lot of the remnant habitat is destroyed, and echidnas may find it difficult to forage, I guess, as widely as they originally could, just while they're trying to avoid predation as well. And so I guess the significance of the most recent bushfire has just presented conditions like we've never seen before. I thought it was really important to understand how this impacts echidnas and their health in order to then, I guess, as a preliminary study, understand whether we need to further delve into this research. So to do this, can you all see my mouse? Yeah, beautiful.
Yeah, perfect.
Thank you. So to do this, a lot of my work actually relies on a citizen science project called Echidna CSI. This was founded by Dr. Talia Perry at the University of Adelaide, and it allows researchers to collaborate with the community by relying on citizen scientists to collect scat samples or poo samples and echidna sightings. So a lot of the samples were found by citizen scientists on Kangaroo Island. So to do this, we had them shipped over to the University of Adelaide. We extracted all the DNA that was present from the scats, and then using PCR amplification, we were able to identify and isolate just the bacteria that are present within the guts of the echidnas. So we did this using the 16S ribosomal RNA gene, which is a highly conserved gene in bacteria. And a core component of this gene is the V4 region. And apologies, it's a bit small on this poster, but the V4 is the largest part of the gene. And so what we did is we basically zoomed into this was able to sequence this DNA and compare it to a library of other bacteria to then identify the species of bacteria that it is. So doing that, we then, as I said, we did the DNA sequencing, and then we did some bioinformatics analysis just to help process the data and turn it into what you see presented at the bottom here. And so I've used some little emojis so it's a bit clearer, but the trees just mean that the samples were collected from before the bushfire, and then we've also got the samples from after the bushfire. So in total, we had 6 successful samples from before the bushfires, 2 from within the fire-affected regions, and then 5 from outside of the fire-affected regions along the eastern side of Kangaroo Island. So what you can see pretty much straight away, which we thought was super interesting, that there's basically a very big shift from before the fire to post-fire. We can see that there's a lot of the proteobacteria and the actinobacteria pre-fire, whereas post-fire there's a lot less of that, and we get more of the Bacteroida and the Firmicutes. So what we thought was super interesting, that there is a reduction in bacterial diversity and composition after the bushfires. Once we did a bit of research, we do also have a local expert on Kangaroo Island. So she was super helpful, just observing their behaviour and their foraging. She did see a few echidnas as well. What we noticed when we dove into the bacteria, so here it's presented at the phylum level, so a very high level perspective. But once we dug into it a bit more, we noticed that the samples from before the fire mostly contained soil bacteria, which we know because echidnas tend to forage a lot, and when they do, they ingest soil at the same time. But when we look at the results after the fire, we found there was a very uniform, a very specific signature, so where the diversity just wasn't as wide as before the fire. And I guess this could be explained because when there is a bushfire, it removes a large proportion of the topsoil. So, and also on top of that, it'll affect soil chemistry aspects like pH, nutrient matter, and organic matter content. And so that removes a lot of the diversity. When we dug a bit further, we found that there were, in addition, a lot of bacteria that matched up with other mammals that consume a lot of ants and termites. So that kind of helped us figure out, so if they've got a very similar microbiome signature in the gut, like maybe they're consuming more ants and termites compared to before the fire, which when you look into it a bit more, ants and termites do quite well at surviving through wildfires. So this made a lot of sense to us. It kind of gave us a bit more information that their diets are quite restricted at the moment. Something that I think is really interesting to point out as well is we do have the one outlier here, where there was a scat sample from an individual echidna that looks a lot similar to those pre-fire. And so when we look at it up in the map here, we actually found the scats on the eastern side of the island, where it's the furthest away from the fire impacted regions. And so we know that echidnas can forage to up to 88 hectares. Up to 88 hectares is their range, and so we thought this individual must have otherwise been less impacted compared to the rest of them. So in summary, we have found that bushfires do really have a significant impact on the gut microbiomes on echidna health. We're not too sure at the moment whether this is a positive or a negative impact, but generally in other studies, we have seen that severe changes in the microbiome can lead to poorer health just because there's a reduced diversity of bacteria present. But basically, this has just led us to believe that we do need to do more comprehensive research into monitoring and planning for our native wildlife. This is more important now than ever, particularly with the current climate that we're in. We can expect longer and drier summers. So yeah, I think this is really cool. This is the first research that I know of that's actually looked at the impact of bushfires on a non-human mammal. So there's some really promising work in there, and can't wait to hear some of your questions. Thank you for listening.
Thank you, Amy. That was super interesting. I love the title. So if people who are listening, if you'd love to put your questions in the chat, I prefer chat to the Q&A box, but if you do either, it's fine. In the meantime, I'd love to hear from our judges if they have any questions for Amy.
Hi, Amy. Michelle here. This is not my field at all, so I really appreciated how you articulated in a way that I could understand, so thank you for making a very conscious effort to do that. I was curious about the samples themselves, how many you had, and could you be sure that they're from different animals, or is that a very green question, so to speak?
No, that's a really valid question. We did start off with about 23 samples, but just because there's quite a few steps with extracting the DNA, getting the sequencing from the DNA, making sure that actually works out, we only really ended up with 13, unfortunately, that actually made it all the way through the entire process. I guess in response to your question about how can we be sure it's an echidna scat, they are very distinct in the way that they look. We only actually had 2 people send in lots of different samples. One of them is an expert in echidnas. So there is the possibility it could also be from not from an echidna, but we can kind of gauge that once we do the DNA sequencing. There's a lot of cleanup, there's a lot of analyzing that goes on behind the scenes to make sure.
And would you be able to tell from your analysis whether this scat was from the same animal, as in like literally that same, I was going to say same person, but no, not same person, same animal?
So it's outside of the scope of my research, just because I only extracted the bacterial DNA.
Yeah, okay. And it's not the same. So your bacterial DNA it doesn't necessarily
to the echidna DNA. Yeah.
Yeah, gotcha. No, thank you very much.
No worries. Thanks for the question, Michelle.
Yeah, I was thinking similar questions, Michelle, so I'm glad you asked it. Sam, did you have any questions for Amy?
Yes, sir. So I have 2 questions. Thanks, Amy, for a nice presentation. It's very clear from this poster as well as with your presentation. So first question is, are you planning to check the microbiome of echidna in other fire affected areas, like in Eastern Australia? This is my first question. And the second question is, are you guys also planning to see the microbiome of the ants that echidna are feeding?
I'm very glad you asked, because the answer to both of those questions is yes. So because our study had some really cool results, it actually inspired one of our research colleagues. I think she's in Melbourne. Her name's Michelle Shaw, and she will actually start to be, because they've got Echidna CSI is an Australia-wide project. So they've got a few samples over in Melbourne that they can use before and after the bushfires, and they've seen that there was indeed a change after the bushfire. So that's inspired them to start their own projects. The research team that I'm currently a part of, we're actually applying for a grant to then be able to let us sequence the DNA of different ants and termites and kind of narrow down the search a bit more, and then can be able to compare that to the results that we got from my research. Does that make sense? So we'll be looking at actually a bit more about the diets and the insects and everything that they'll be eating, so we can definitely confirm, even maybe down to the species level, depending how far we get into that.
Thank you.
Yeah.
Awesome. It's so good that you have such a big team. This recording will be made available online following the conference, and of course we hope to see you in other sessions. So thank you.
Great job. Nice to meet everybody.
All the best with future research.